Okay, folks, so we just got finished talking about our amino acid, and I want to show you that there are actually only 20 different amino acids. So all the protein in the world is only like made up of, these building blocks, these amino acids. Okay, remember that's like the monomer. Okay, so this whole thing is one little bead, okay? But a whole protein would be the polymer version, would be this. So all of these beads are only 20 different amino acids. So it'd be nice if I had 20 different colors and I could show you like there's just 20 different colors of that. But proteins can be very long, very short, whatever. Okay, so I want to show you those 20 amino acids, and you do not, please do not copy all of these. Okay, you do not need to know that. Okay, so here we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. 20 different amino acids. And you'll notice a lot of them end in INE. Okay, INE. Some end in an acid because it's a amino acid, okay? And the INE stands for the amine kind of thing, okay? But they will become proteins if you link all these things together, and you can link them in any kind of order. But what I want you to see is because they're amino acids, they all have those 4 components around a central atom. So let's just jump to like this one right here. This is called cysteine, okay? And here's your central carbon. Let me get another color and hopefully these things will kind of stand out a little bit more. Okay, off to the side here, here's that N with 2 H's. That's the amine group, amine. And then off to the other side is the carboxyl. That's the C double bond, and then the OH. That's the, I circled it on the wrong one here. I'm going to circle it on this one over here. That's the carboxyl. Okay, up here at the top is just the hydrogen, okay, because remember they have an amine group, they have a carboxyl group, they have a hydrogen. Then everything else from there on down, one more color here, okay, everything else from here on down, I'll put it in like this kind of thing. That is the R group. That's what makes every single one of them different. And if I go around and I kind of box or, outline all the R groups, you can see they all differ. Some are very big, some are just a little bit, okay? Some even have this like funky kind of thing down here going on, okay? Some even, their R groups kind of connect with their amine group, okay? That's like a weird one there. Here's another one down here. Here's another one. It's a crazy R group is all this. Oh, missed that hydrogen in there too. Okay, but you can see that the 20 different amino acids, they all have the amine and the acid. Technically, that's where they get their name, amino acid. They all have a hydrogen, and then they have the R group. Now, these 4 things don't always have to be in that location. Like the amine group could be at the top, and the R group could be over here, and the H could be down here, but you will always have those 4 things. It doesn't matter where they're positioned around that central carbon, but you will always have those 4 things around: the amine group, the R group, the hydrogen, and the carboxyl amino acids, okay? So those are our monomers, excuse me, okay? And one more time, I'll put back, the monomer is the amino acid, right? But the polymer, okay, now this is kind of tricky. We want to say the polymer is called a protein, right? But actually, it's not a protein till it becomes a certain shape. They call it first a polypeptide. All right, polypeptide. And this is where you can spell this, polypeptide. Okay, you like that word poly. I know you do. It means many. But why didn't it say polyamino acid? Like polysaccharides, from monosaccharides, polysaccharides. And why didn't they call amino acids monoamino acids? That would have been nice because it would have made a lot more sense. But we're going to see where this peptide comes from. Okay, so let's take a look at this over here. We have 2 amino acids. They happen to be the same type. They have an H, but it happens to be that their R group is an H. And I think if I pulled that picture of all the amino acids, one of them, there it is, glycine, is the one that has an H, and then its R group is also just an H. So these are 2 glycine amino acids. And what we're going to do is we're going to link them together. Remember how we would take one monomer plus another monomer, right? And when we link them together, remember that? We have to remove water, and that's called dehydration synthesis. Well, that's what's happening here. You can see that this OH and this H, that's H2O, right, gets removed. Whoop, out it comes. And that's what connects them. And then this bond that forms where these two were, these two like form a bond across here, that is called the peptide bond. Ooh, remember polypeptide? This is why we call the polymer form polypeptides, because it will have many peptide bonds, because you won't have just two. You'll have many, many amino acids like linked. So it'll be a polypeptide. But when does it become a protein? Well, kind of like, when do you guys become an adult? It's when you've actually like formed you. Okay. Like right now you're growing up, you're trying to find yourself, but a protein isn't a true protein. It's just a polypeptide until it becomes like something else. And so what we're going to do is we're going to look, and this is some extra information. So that's it on polymer form, by the way. Peptide and protein, you can use those like synonyms. But what I'm going to show you is the shape of proteins. Okay. How does a polypeptide become a protein? Let's write that down. How a polypeptide becomes a protein. Okay, and I'm going to show you how that happens, okay? And it happens in kind of two steps. And what I have here to like assist me, my lovely assistant here this time, is not a chain, okay? Instead, I'm going to say that this ribbon is like a chain. This is gift wrap ribbon, okay? And you see all the ridges on it? Imagine each one of those ridges is an amino acid. So this would be lots of amino acids. This would be bigger than this chain is, right? This would be lots of amino acids, right? And the sequence of those amino acids, like whether this first one be like glycine or the next one's leucine, all those 20 amino acids, could be all 20. It could be a whole bunch of them right in a row or something. That sequence of amino acid is what we call the first like kind of shape in becoming a protein. And we call it the primary structure. Okay, and that primary structure is the sequence of amino acids. The sequence of amino acids. And that will be dictated by your DNA. Like every protein in your body is like built, excuse me, things are just falling around here, is built based from your DNA. Your DNA kind of says what is the sequence of amino acids. That information will come directly from your DNA. Okay. Now that sequence of amino acids might not mean much. Okay. It's kind of like, imagine having this ribbon is just like that. So we have to go into the next kind of thing. Like what's the next development of a polypeptide into a protein? Well, we call it the secondary structure. And by the way, primary is symbolized with 1 with like a little degree. So secondary is 2 with a little degree. And the secondary is one of two things is going to happen to that sequence. It can either, and I need a pair of scissors. Grab a pair of scissors out of my drawer here. I need a pair of scissors to illustrate this. How many times have you ever wrapped a gift and then you go like this with the gift stuff there? Whoa, what? Whoa. Here, let me get some more here. Okay, you take this, you take a pair of scissors on this and you drag it across those ridges, right? Cool sound, right? And you get this coily-Q thing, right? Okay, well, that's what proteins do in the secondary structure, okay? That's one way they might do it, okay? That's one way they might do it. Another way they might do it is what we call pleats, okay? You know what pleats are? You might have blinds in your house or something on your windows that are pleated, or you might have pants that have like a pleat at the waistline, or you might have a skirt that has pleats. Like Scottish people have pleated kilts all the time. Okay, pleats are like this zigzag form. Okay, so proteins are going to do one of two things in the secondary structure. They're either going to be coils or pleats. Okay, so we have one of these kind of representing the coils and one of these representing the pleats. Okay, still not a protein yet. It's still got to develop a little bit more. Okay, so what are we going to get next? What's the next shape we're going to get? Well, primary, secondary. What's the third thing called? It's called a weird word called tertiary. Tertiary structure. And I'm not going to write structure. I'm just going to write the little symbol. And tertiary structure, I like to say, is globs. Globs. So we're going to take this nice coil that I just did, and we're going to glob it up. Glob it up. Okay. Some of the coils you can still see are still there, but now it's all like globbed together. Let's glob this little tail part together too. Okay, that is a glob. We're going to do that to this one too. We're going to glob. This is kind of harder to glob it together. And you can still see some of the pleats there, okay? That's the tertiary structure of these two chains of amino acids, right? Because we had the primary, the sequence of amino acids. We either coiled it or we pleated it. Now we globbed it up. Finally, the fourth, which is called quaternary, you can call it just the fourth level or 4 with a little degree, is what I call globs on globs. Globs on globs. Okay, when that happens, it's like kind of like you take these two things and we're going to like kind of tie them together or something. Okay, so we're going to glob the two things together. When you got that, you have a very specific shape of a protein. Now, I know that shape doesn't look pretty or anything, but your body will build that shape very specifically because that shape is going to matter. That shape is going to be like a lock. And just like a certain key has to fit into that lock, that has to be shaped exactly like it should be. And if it's not, it's totally destroyed. So where does shape matter? And this is the second thing I want to talk about with extra info on proteins. Where does shape matter? It matters with enzymes. Okay? Enzymes. Enzymes, remember we said, speed up reactions in your body. Okay, so your metabolism can be sped up by this. I'm going to draw an enzyme, and I'm going to imagine it like drawn like this, right? And imagine this enzyme is like the key. The enzyme could be the lock or the key, by the way. Okay, imagine this is the enzyme, right? And it's got to fit into a specific lock. So that lock's got to look like this, right? Okay, this lock will be called the substrate. The substrate. And this substrate could be, say, lactose. Okay, and if you have the enzyme lactase, enzymes always end in ase, by the way. You have the enzyme lactase, then it will fit in with lactose, just like this. I hope I don't run out of time to show you this as I draw it all. Will fit together, and then when it comes apart, the enzyme remains unchanged, okay, and can be used over again. But the substrate now is broken down into this and this. This is glucose and galactose, or your products. This is if you make the enzyme lactase, right? Then you are lactose tolerant. You can then digest lactose. And if you have that enzyme, when it comes together with the lactose, it breaks it down to glucose and galactose. It digests it for you. But if you don't have that enzyme, the lactose would stay like that in your body and it would probably spoil. Okay, it would make you very sick. So enzymes speed up reaction. There's lots of enzymes out there, and we may even do a little activity on them. And that's it for proteins, you guys. Our final category will be nucleic acids, and that'll be in the next video. Bye.